In the late 1980s, inside a sterile laboratory in Japan, chemists hunched over molecular models. They weren't creating something from nothing, they were mimicking nature's own defense. Their target was nicotine, the potent alkaloid that tobacco plants evolved to kill insects. The goal was to capture its lethal efficiency, but in a new, synthetic form that could be manufactured at scale. Their hands moved flasks and pipettes, combining precise reagents under the hum of fume hoods. The process was one of deliberate molecular surgery, tweaking the nicotine structure to enhance its insect-killing power while aiming for selective toxicity. They synthesized a series of crystalline compounds, each variation tested on trays of aphids or beetles, watching for the telltale paralysis that signaled a successful hit. The most promising molecules shared a core trait. They were neurotoxins designed to overwhelm an insect's nervous system. They bound irreversibly to specific receptors in the pest's brain, triggering a fatal flood of signals. This new class was christened neonicotinoids, new nicotine-like insecticides. In that quiet lab, a tool was forged that would soon be scattered across global farmlands. The chemists had created more than just a new spray. They engineered a poison that could be woven into the plant itself. Farmers began receiving seeds pre-coated with the chemical, tiny grains of corn or soybean encased in a thin, toxic shell. They planted them as usual, burying the poison with the promise of growth. As the seed germinated, the neonicotinoid dissolved into the seedling's first sip of water. It hitched a ride in the xylem, the plant's internal plumbing, and spread upwards with the rising sap. The toxin permeated every new leaf and stem, turning the entire growing plant into a passive, poisonous shield. This systemic design meant insects chewing on any part of the mature crop would ingest the chemical, but the poison didn't stop at the leaves. It travelled further into the plant's reproductive systems, eventually reaching the pollen and nectar within its flowers. The defense mechanism was now complete and utterly invisible. The innovation that began in Japanese laboratories now spread across entire continents. By the 1990s, millions of acres of North American and European farmland were sown with seeds sheathed in that same synthetic nicotine. The chemical's systemic promise was fulfilled. A farmer needed only to plant the coated seed once. As spring turned to summer, vast fields of sunflowers and canola erupted from the soil, their leaves unfurling a vibrant, deceptive green. From the air, these agricultural landscapes appeared as perfect mosaics of health. The flowers bloomed on schedule, their yellow and purple heads nodding in the breeze, showing no outward sign of the toxin woven into their very fibers. The system seemed flawless. The poison travelled upward from the root, permeating stem and leaf, creating an invisible shield against pests that chewed or bored into the plant. For the grower, it was a season of quiet confidence, watching a protected crop mature without a single sprayer leaving the barn. But the chemistry that defended the plant did not discriminate. The same vascular system that carried water and nutrients also distributed the insecticide into the pollen and nectar at the flower's heart. There, the poison waited, not as a visible residue, but as a fundamental component of the plant's own offerings. The field was a trap in full bloom, its beauty a perfect camouflage for the neurotoxic payload hidden within each grain of pollen and every drop of sweet nectar. The sunflower field from the previous chapter, now in full bloom, hummed with life. A honeybee forager, drawn by the bright yellow petals, landed on a flower head heavy with pollen. Its legs sank into the powdery yellow dust, and its proboscis dipped into the nectar reservoir at the flower's centre. Both resources, unbeknownst to the insect, carried microscopic traces of the systemic insecticide that had travelled up from the treated seed. On its hind legs, the bee's pollen baskets, tiny concave hairs fringed with longer bristles, began to fill. It methodically packed the contaminated yellow grains into moist pellets. Meanwhile, in its honey stomach, a separate internal sac, it stored the tainted nectar. The process looked utterly normal, a ritual performed millions of times a day. Yet with every movement, the bee was consolidating a poison payload for transport. Its foraging complete, the bee lifted from the sunflower, its legs weighed down by the full pollen baskets. It oriented itself toward the hive, a journey of perhaps several kilometers. The insect flew a straight, determined line, carrying the contaminated resources home. There was no outward sign of distress. The toxin was hidden within the very food meant to sustain the colony. The contaminated pollen, stored in hexagonal cells, became the colony's next meal. Nurse bees, their glands secreting royal jelly, mixed this tainted harvest into the brood food. They passed it mouth to the pale, grub-like larvae curled in their wax cradles, unaware they were administering a slow-acting poison with every feeding. Inside those developing larvae, the neonicotinoids began their subtle sabotage. The chemicals bound to receptors in the young bees' nervous systems, disrupting the signals that governed growth. Studies later revealed larvae fed this diet often emerged smaller, with underdeveloped wings or malformed bodies, their fundamental architecture compromised from the very start. The damage ran deeper than mere deformity. The poison suppressed key genes responsible for immune response. A bee born from this contaminated nursery entered the world with a crippled defense system, far more vulnerable to common hive parasites and pathogens that a healthy colony might otherwise withstand. Back at the hive, the returning forager's disorientation was just the beginning. Scientists in a French laboratory had glued a speck of a tracking tag, a dot smaller than a pinhead, to the back of a bee that had fed on tainted sugar water. They released her into a flight arena, a white dome crisscrossed with infrared beams that mapped every turn and hesitation. Her flight path, rendered on a screen as a jagged red line, told the story. Instead of the confident, looping arcs of a healthy bee navigating back to a virtual hive, her trail wobbled and doubled back on itself. The toxin had scrambled the neural circuits for spatial memory. She could not recall the simple route she had learned just hours before. In the real world, this meant a forager might circle a familiar hedgerow, utterly lost as dusk fell. Her energy would drain, her wings grow heavy. She would never deliver her cargo of poisoned nectar, and she would never return home. The hive, piece by piece, was losing its navigators to a chemical fog. The disoriented foragers never returned home. By the mid-2000s, American beekeepers began lifting hive lids to discover a haunting new reality. The queen remained, alive and moving, but her court had vanished. Only a few confused workers circled her, their buzzing thin and desperate in the vast hollowed-out wooden box. The rest of the colony was simply gone. The beekeepers' hands moved through the silent frames, finding them heavy with untouched honey and pollen stores. Brood cells that should have held developing larvae were eerily empty or capped over with dead pupae. There were no piles of dead bees at the entrance, no obvious sign of a predator or disease. It was as if the workforce had collectively walked off the job, abandoning their food, their young, and their queen. Scientists documented the pattern and gave it a name: colony collapse disorder. The symptom was a catastrophic failure of the social contract. A healthy hive is a buzzing metropolis. This was a ghost town with its lights still on. The mystery wasn't just death, it was disappearance. The poison had not necessarily killed the bees in the field. It had severed the invisible threads that bound them to home. While beekeepers in America were finding their hives eerily empty, scientists across the Atlantic were designing a different kind of experiment. They wanted to measure not just death, but decline. In the English countryside, researchers placed dozens of bumblebee colonies at the edges of vast, yellow-flowering fields of oilseed rape. The crop had been grown from seeds coated with the same systemic insecticide. Throughout the spring and summer, the team returned weekly. They would carefully lift each wooden nest box, its weight telling a silent story of the colony's health. A thriving bumblebee colony should grow heavy with new workers, pollen stores, and the future of the species, new queen bees, fattening in their wax cells. But the scales told a different tale. By season's end, the data was stark. Colonies near the treated fields were significantly lighter. More critically, they had produced a fraction of the new queens compared to colonies placed near untreated meadows. The pesticide wasn't just killing individual bees, it was strangling the reproductive cycle of the entire colony, quietly ensuring there would be no next generation to found new nests the following year. As evidence mounted from both field studies and laboratory experiments, the debate shifted from scientific journals to government corridors. By the early 2010s, regulators at the European Food Safety Authority began sifting through thousands of pages of toxicological data. Their task was monumental: to determine if the benefits of these systemic insecticides still outweighed the documented risks to pollinators. In sterile laboratory rooms, scientists conducted the final, definitive tests. Using fine syringes, they fed individual honeybees precise microliters of sugar water laced with neonicotinoids. They watched for the subtlest signs: a delayed reaction to light, a stumble in flight coordination, meticulously recording the dose at which harm began. This data would form the bedrock of new safety limits. The culmination arrived in 2013. Citing an unacceptable risk to bees, the European Union voted to severely restrict the outdoor use of 3 major neonicotinoids: clothianidin, imidacloprid, and thiamethoxam. The ruling sent shockwaves through global agriculture, creating a stark regulatory divide and forcing a fundamental re-evaluation of how to protect crops. As the regulatory debates of the previous decade settled into policy, agriculture entered a new phase of cautious coexistence. In regions where neonicotinoid restrictions took hold, farmers began adopting integrated pest management, a system that combined crop rotation, beneficial insects, and targeted spraying only when pest thresholds were exceeded. The approach required more scouting through fields, more patience, but promised to break the cycle of prophylactic chemical dependence. Yet in other parts of the world, the coated seeds remained standard practice. A soybean farmer in Brazil might still plant neonicotinoid-treated rows, the chemical travelling upward with each new leaf. The fields appeared productive, the harvests robust, but beneath the apparent success, a silent exchange continued. Pollinators visiting flowering cover crops or adjacent wildflowers still carried microdoses back to their nests. To measure the hidden toll, beekeepers turned to precision scales. They placed entire hives on electronic platforms that logged weight changes hourly. A stable or rising curve meant successful foraging. A sudden drop could signal disoriented bees failing to return. This constant digital vigil became the new normal, a numerical heartbeat tracking a colony's health in a chemically complex world.